osmosis, that is the very important thing in case for the bacteria. This is what? A diffusion of water from high concentration to the lower concentration, right? You have studied, no, this kinds of the experiment where the water molecule will go from the lower concentration to the higher concentration of the osmosis, right? So with the help of these experiments, we can study the three different kinds of the condition in case of the cell, where the water molecule will go from outside to inside, means the inside the cell the water molecule will be increased, and the condition will be what? Lysis of the cell. If there will be balance of the water molecule inside and outside of the cell, then it is the normal condition. And if the water molecule will go inside to the outside, means in the environment, then it will what? Shrinking of the cell. So this is generally happen in what? Salt water. In the fresh water, either cell will be make a equilibrium, means the normal condition, either cell will lyse in case of the fresh water, but in case of the salt water, bacteria will always transport a water molecule from inside to the outside, and there will be a shrinking condition. So this is the major structure of the bacterial cell. After plasma membrane, this is the cell wall, that is the major portion of the bacterial envelope that determines the cell shape, that prevents the lysis from the cell, and it is made up with the help of the major portion that is known as what? Peptidoglycan. There are two different kinds of the prokaryotic organism, that is known as what? Gram positive organism and gram negative organism, that can be differentiated on the basis of what? The amount of the peptidoglycan on the cell envelope, right? If we will consider the gram positive cell wall, then that is the thick homogeneous sheath of the peptidoglycan, which is made up with the help of 20 to 80 nanometer of the thickness. So if we will compare the negative and the positive organism, the positive organisms are having the higher concentration of the peptidoglycan. Now along with the peptidoglycan, the gram positive cell wall also contains the teichoic acid as well as the lipoteichoic acid. Now what is the function of the teichoic acid and the lipoteichoic acid? The function in the cell wall maintenance and the enlargement during the cell division. As we know that the prokaryotic cell division will be carried out by the binary fission only, right? If we consider the eukaryotic organism, then it is the mitosis and meiosis. But for the binary fission, first organism has to enlarge their cell surface, and after that all the organism will double with the help of the replication process, and all the doubled organelles will bifurcate into the two different portions, and after that one cell converts into the two. So for that cell, at the time of the cell division, it requires what? Enlargement, and this enlargement will provide by the teichoic as well as the lipoteichoic acid. Now this is the structure of the peptidoglycan. Generally it is made up with the help of N-acetylmuramic acid and the N-acetylglucosamine, right? If we will consider the structure of the N-acetylmuramic acid, there are four different kinds of the amino acid. This is what? L-alanine, D-glutamic acid, and diaminopimalic acid, and D-alanine. Four different amino acids are present on N-acetylmuramic acid. This is from this portion the question has been asked that which are the amino acid which is not present in the peptidoglycan. इनमें से इस चार से किसी और अमीनो एसिड देकर ऐसा क्वेश्चन पूछा जा सकता है कि कौन सा अमीनो एसिड पेप्टिडोग्लाइकन का पार्ट नहीं है. सो ये चार अगर आपको आते हैं कि एल एलेनाइन, डी ग्लूटामिक एसिड, पिमेलिक एसिड एंड एलेनाइन इज द पार्ट ऑफ एन एसिटाइल म्यूरामिक एसिड, देन अदर 16 अमीनो एसिड आर नॉट द पार्ट ऑफ द पेप्टिडोग्लाइकन मॉलिक्यूल्स. So in case of the N-acetylglucosamine and N-acetylmuramic acid, there will be a monomer. After polymerization, there will be a structure of the whole peptidoglycan, or we can say a cell wall. There will be a cross-linking between the N-acetylmuramic acid and glucosamine. Sometimes it is also made up with the help of the glycine molecules with the help of the peptide bridges. sometimes it is in a simple molecule, simple bridge. This is the cross-linking between N-acetyl and N-acetyl muramic acid. This is the ultimate structure of the cell wall, which is made up with the help of the peptidoglycan. See over here, this is the peptidoglycan N-acetyl muramic acid and N-acetyl glucosamine, which is cross-linked by the glycosidic bond, and also a presence of lipotecoic and tecoic acid for the enlargement of the surface. This is the structure of the tecoic acid. Now this tecoic acid is having the polymers of the glycerol, or sometimes it is known as the ribitols. Glycerol as well as the ribitol molecules, polymer of glycerol and ribitol with the help of the phosphate molecules. Combiningly it is known as what? Tecoic acid. Now let us start with the gram-negative cell wall. So gram-negative cell walls composed of an outer membrane, thin layer of the peptidoglycan. Outer membrane is similar to the cell membrane of the bilayer structure. This is made up of the lipopolysaccharides and the lipoproteins. Lipid portion may become toxic when released during the infection. It may function as a receptor and blocking in case of the immune response. It contains the porine protein, as we have already discussed in the transportation mechanism. It contains the porine protein in the upper layer, and it regulates the molecule entering into the living cells. Now bottom layer is a thin sheet of the peptidoglycan and the periplasmic space above and below peptidoglycan. Let's see the structure of the gram-negative cell wall. This is the thin layer of the peptidoglycan structure. This is the outer membrane, and this is the inner plasma membrane that we have discussed, no? That the protein molecules are there. So here one more membrane is there, that is known as the outer membrane, and this outer membrane contains the different kinds of the lipopolysaccharides as well as the lipoproteins, right? Now this lipopolysaccharides, so this is the typical structure of the lipopolysaccharide, that is made up of the O side chain, core polysaccharide and the lipid A. So on the basis of O side chain, many a times organism can be differentiated by the serovars. No, we have discussed like morphovars, biovars and serovars. So as the O side chain will differ, the organisms are having the different serovars, though they are having the similar characteristics biochemically or structurally or the function, but serologically they are different on the basis of this O side chain. That is only well studied in the Salmonella organisms. Now this is the comparison between the gram-positive and gram-negative cell wall. As we can see that gram-positive contains the thick cell wall, while the gram-negative contains the very thin, but gram-negative contains two different kinds of the membrane, that is outer membrane and the inner cell membrane. This is the major comparison between the gram-positive and the negative. While in case of the gram-negative, this is the outer membrane, contains the porine protein, phospholipids as well as the lipopolysaccharides. So these lipopolysaccharides are having sometimes antigenic activity. Porine protein channel have the selective permeability, that is lack in the gram-positive bacteria. So gram-positive and gram-negative, if we will compare the archaeal cell wall, then again there is some variation into the archaeal cell structure. There is examples of the Methanobacterium as well as the Thermoproteus. There is a cell surface here, the cytoplasmic membrane, this is the cytoplasm, right? So here in case of the gram-positive archaea, there is a thick cell wall made up with the help of the peptidoglycan, but there is an absence of the peptidoglycan layer in case of the gram-negative archaea. So there is again a difference in the bacterial cell wall, gram-positive and negative, as well as the archaeal gram-positive and negative cell wall. In case of the archaeal cell wall, that is made up with the help of pseudomurine. In case of the bacterial cell wall, that is made up with the help of what? Peptidoglycan, and the monomers are N-acetyl glucosamine and N-acetyl muramic acid, where here in case of the archean that is made up with the help of pseudomurane, that the monomers are N-acetyl seminoic acid and N-acetyl glucosamine. So glucosamine is the similar one, but here the instead of N-acetyl muramic acid, the different molecules will be there. So this is the major difference between gram positive and negative organism. Number of layers in case of the gram positive, only one. In case of the gram negative, 2 layers are there because it contains 2 membranes, one cell wall. Chemical composition of the gram positive, that is made up of peptidoglycan, teichoic, lipoteichoic acid, mycolic acid and sometimes polysaccharides. Where in case of the gram negative, that is lipopolysaccharide, lipoproteins, peptidoglycan and the porine protein, which is absent in the chemical gram positive. Porine proteins are not there in the gram positive bacteria. Overall thickness of the gram positive bacteria, that is 20 to 80 nanometer, thinner here in case of the gram negative, thinner up to 8 to 10 nanometers. Outer membrane is not there in the gram positive, whereas outer membrane is positive in the gram negative bacteria. Periplasmic space, that is narrow in case of the gram positive, extensive in the gram negative. If we talk about the permeability of the molecules, that is more penetrable in case of the gram positive, while in case of the gram negative it is less penetrable because they are having the 2 membrane, outer membrane as well as the inner membrane. And on the basis of the cell wall composition of the peptidoglycan, the organism can stain into 2 different, that is gram positive and gram negative, gram stains, right, that you have studied in the earlier semester, no? Gram reaction, the addition of the first thing, that is what, crystal violet, then you allow to react with the cell wall, then wipe out the crystal violet, add the iodine as a, iodine will act as a what? Mordant, right? That, what is mordant? What is the function of mordant?
In the binding of the dye.
That will make a more specific binding of the dye with the cell wall, right? So after that you will add the, after iodine, there will be a addition of the crystal violet, and after that the cell will become, differentiate the cell gram negative or the gram positive. So this is the mechanism by which you can differentiate. Those are a gram positive, they are having the thick peptidoglycan layer, and when you will apply the crystal violet, there will be a specific binding with the cell wall with the crystal violet, and at last it will not colored with the safranin, that's why it will remain what, blue in color or purple in color, while due to the thin layer of the peptidoglycan, CVI complex, crystal violet iodine complex, will remove and counter stain, which is the safranin, that will stain the gram negative bacteria, and that's why the gram negative bacteria remains the pink in color, right? So the cell wall composition of the peptidoglycan, that will play a major role over here to differentiate the organism into the 2 different categories, like the gram negative and the positive category. There are some non-typical cell walls are also there, that is what, some bacterial groups that lacks the typical cell wall of the peptidoglycan, that doesn't contain this kinds of the cell wall. There the examples are the Mycobacterium and the Nocardia. Gram positive cell wall structure with the lipid mycolic acid, and this kinds of the microorganisms having the pathogenicity and the high degree of the resistance to the certain chemicals and the dye, and that can be used for the acid fast stain. So for example, in case of the Mycobacterium, there is a lack of the typical kind of the peptidoglycan cell wall. And another one is known as what, Mycoplasma, that is cell wall less organisms, and due to the absence of the cell, they are not having a particular kind of the cell shape, as we know that the cell wall provide the shape to the organisms, and that's why the absence of the cell, the organism is known as what, pleomorphic. Pleomorphic means at different times cell are having the different types of the shape, that is known as the pleomorphic. The another portion is the cell cytoplasm, that is known as the dense gelatinous solution of the sugar, amino acids and the salt. It contains almost 70 to 80% of the water, and it is served as a solvent for the material used in all the cell functions. So within the cytoplasm, it contains the chromosome. If we will consider the prokaryotic genetic material, then it is only a single circular double-stranded DNA molecules which is scattered throughout the cytoplasm. There is no particular kind of the boundary, and that's why it is known as what? Nucleoid, right? Where in case of the eukaryote, they are having the particular kind of the boundary, and that is known as the nucleus. Another one is known as the bacterial internal structure, that is plasmid. What is plasmid? That is extra chromosomal DNA, right? That is small circular double-stranded DNA, free or integrated into the chromosomal DNA, right? Duplicated and passed into the offspring. They are self-replicating, means that is not dependent on the replication of the main chromosomal DNA of the microorganism, self-replicating, and after the replication of the plasmid DNA, that will pass on to the offspring. Not essential to the bacterial growth and the metabolism. There is no particular requirement of the particular function for the growth, particular metabolism of the organism. It may encode antibiotic resistance, tolerance to the toxic metals, enzymes and the toxins. We have studied regarding the R plasmid, right? What is R plasmid? R stands for what? Resistance. R stands for the resistance, right? So in that plasmid, the numbers of genes are there. That gene represent for the antibiotic resistance, gene represents for the tolerance to the toxic materials, tolerance against the metal or some enzymes or other toxins, right? So if cell will expose by the different types of the antibiotic or heavy metals or the hazardous metal or hazardous toxins, if organisms are having the plasmid contains the resistance gene against that, then the organism can survive in the exposure of the antibiotic or other hazardous material. Otherwise, in exposure of these kinds of the metals or the antibiotics or the other toxin molecules, organism cannot survive. So this is the beneficial part of the plasmid, that contains the resistance factor that can help the organisms to grow in the particular condition. That is also used in the genetic engineering, that you have studied the recombinant DNA technology, right? The organisms can, that plasmid can used as a vector. A particular fragment of the gene can insert into the plasmid, that is known as now recombinant plasmid. That recombinant plasmid can enter into the host cell, and now this host cell can produce the particular product which is encoded by the gene, which is transported via plasmid. So plasmid can also use for the genetic engineering purpose. Then ribosomes, yes? F and HFR, that is generally used for the conjugation, right? F and HFR are having the two different kinds of the characteristics. One is known as the recipient and one is known as the donor one, right? Now generally a pilus can be used for the conjugation. See, there are three different kinds of the gene transfer mechanism. That is what? Transduction, transfection and the conjugation, right? That is the gene transfer mechanism. In case of the transduction, direct gene fragment can be passed from outside to inside the cell. Transformation, that is via the bacteriophage, and the conjugation, that is between a two organism. Pilus can be form one tube-like structure, and from one to another organism, particular characteristic which is absent into the recipient, that will transfer to from the donor. So HFR and H plus can be used for the conjugation purpose. So another one internal structure, that is the ribosome, that is made up of the 60% ribosomal RNA and 40% of the protein, that consists of two subunit, large and small subunit. Now prokaryotic and eukaryotic, earlier we have discussed that prokaryotics are having the 70S ribosomes while eukaryotics are having the 80S ribosomes. Why it is called 70S and 80S? What stands for the S? S stands for the what? Swedberg unit, and Swedberg unit is what? a particular sedimentation rate of that particular molecule. So on the basis of that, a molecule can be defined as a 70S molecule and the 80S. 80S in case of the eukaryotes. Prokaryote contains what? 70S. But if we'll consider this 70S ribosome, then it is made up with the help of two molecules, 50S and the 30S. It doesn't mean that the 50 plus 30 is 80. It is what? 70S ribosome, because on the basis of the Svedberg or the sedimentation rate of this particular 70S ribosome of the prokaryotic structure. So the function of the ribosome is what? That is used in the protein synthesis, right? So this is the subunits of the prokaryotic ribosome. It is made up with the help of 70S, like two molecule larger subunit, that is 50, and the smaller subunit 30. Another intracellular are the inclusion bodies, or we can say granules. In case of the granules, there are two different kinds of the granules, organic granules and the inorganic granules. Now granules or inclusion bodies are generally used for the act as a energy reservoirs, right? They reserve the energy in a form of carbon, in a form of sulfur or the phosphorus. In case of the organic granules, the examples are the glycogen molecules or glycogen granules, or the poly-beta hydroxy butyrate granules. Sometimes it is the gas vesicles, and in case of the inorganic molecules, they are a sulfur as well as the phosphate granules. We have studied regarding the metachromatic granules. What is the composition of the metachromatic granules? That is made up with the help of what? Yes, phosphorus, inorganic molecules, right? So the major two different granules are there, inorganic as well as the organic, but the purpose is what? To reserve the energy. Cytoskeleton. Very poor cytoskeletons has been studied in the prokaryotic, while eukaryotic cell are having the well developed cytoskeleton. But in case of the prokaryotic organism, actin filaments are there, which is used as a cytoskeleton. Many bacteria possess the internal network of the protein polymer that is closely associated with the cell wall, right? So that prepare the cytoskeleton with the help of the actin filaments. This is regarding the bacterial shape, arrangement and size that we have already completed earlier. External structures. In case of the external structures, different appendages are there. There are two major appendages, that is the flagella and the pili as well as the fimbriae. Flagella can be used for the motility, and fimbriae and pili can be used for the attachment or for the channel. And the glycocalyx is the another one outer surface or external structure of the cell that is used for the surface coating. Now if we'll consider the flagellar structure, that is made up with the help of the three major parts, that is filament, or filament is made up with the help of the different kinds of the proteins like flagellin protein, second one hook, and the basal bodies. Let us see the structure of the flagella. According to the cell wall structure, there are two different kinds of the structure of the flagella. In case of the gram positive, thick peptidoglycan is there. Here the gram negative organism, thin layer of the peptidoglycan, but the two outer membrane and inner membrane will be there. So accordingly, the structure of the flagella will be differ in case of the gram negative and the gram positive cell. So here if we will consider, this is known as the hook, that is similar in both the flagella, gram positive flagella or the negative flagella. This is the basal body. So the basal body is different in both the organism. Here there will be four different kinds of the ring in the basal body of the gram negative bacteria, while only two rings are there in the basal body of the gram positive bacteria. Now the basal body are having the four different kinds of the ring, that is known as the L ring, P ring. L ring represent the near the lipopolysaccharide of the outer membrane. P rings near to the peptidoglycan layer. MS ring and the C ring. Two different kinds of the proteins are there, that is known as the moat protein and the flea protein. Now the major functions of the flagella, that depends on the moat protein and the flea protein. The flea protein will act as a motor switching on and switching off. Once the flea protein will be activated, means the motor will be switched, moat protein will be activated, and once the moat protein will be activated, flagellar protein, which is present over here on the hook, that will be activated and that will convert into the movement, right? So the main two proteins are responsible for the movement of the flagella, that is moat and flea protein. Flea will act as a switching on and off protein. So this may be the ask as a MCQs in the maybe in the examination, that which is the responsible for the switching on and off process of the flagellar movement. Now on the basis of the arrangement of the flagellar, there are four different kinds of the arrangement, that is known as the monotrichous, lophotrichous, amphitrichous and the peritrichous. Monotrichous means the single flagellum at the one end. Lophotrichous means the small bunches emerging from the same site. Amphitrichous, that is flagella at the both end, and the peritrichous means flagella dispersed over the surface of the whole cell. This is the examples of the monotrichous. This is lophotrichous and this is what? Amphitrichous, right? And the lophotrichous means if the flagella is the presence of the flagella on the whole cell, whole surface of the cell, then it is known as what? That is peritrichous. Now on the basis of the flea and moat protein, the movement can be divided into two, that is known as the running and tumbling. The flagellar movement has been divided into two, running and tumbling, but that is on the basis of again a clockwise movement and the anti-clockwise movement, right? If the flagellar protein, flagellin protein, which is present in the hook here, that will rotate anti-clockwise, then the microorganism, the cell can easily run, that is known as the running. But if the flagellin protein move, or flagella, whole flagella, thread-like structure will move in clockwise, see this is the clockwise structure, right? Then it will be known as what? Tumbling. In case of the running, that is anti-clockwise, there is the whole bunch together and make some force, that is the running motion. But in case of the clockwise motion, there is no bunch-like structure of the flagella, and that's why there is no force to the cell. Cell will remain move at the same space, that is known as the tumbling, right? So this is known as the tumbling and the running process. Now in case of the particular attractant molecule or the repellent molecule, cell move ahead. See over here, in case of the condition A, there is no attractant or the repellent, then in case of the tumbling, the cell will remain the same place. Or in case of the attractant, presence of attractant, in case of the tumbling, cell will remain at the same place, but in case of the running, they will move from one to another place. So for the running purpose, the particular kind of the attractant molecule should be there. So the anti-clockwise molecule, anti-clockwise motion will be carried out of the flagellin protein, and the running process will be carried out. Sometimes a periplasmic flagella is also there, that is present in the spirochete organism. Periplasmic means there is no outer flagella, but there is the presence of flagella between the periplasmic space. That is the example in the spirochete, right? Spirochete contains the periplasmic flagella, that is also responsible for the helical movement of the spirochete organism. Another one outer structure is the fimbriae, that is the fine proteinaceous hair-like structure, that is known as the fimbriae, and the main function of the fimbriae is the adhesion to the other cells or the other surface. Pili, that is rigid tubular structure made up of the pili protein, found only in the gram-negative cells, functions of the pili is to join the bacterial cell for the partial DNA transfer, right? As we have discussed, there are three different kinds of the gene transfer mechanism: transduction, conjugation, and the transfection. So the pili can be useful in the conjugation process, which is the DNA transfer from one bacteria to the another bacteria. So here in case of the bacteria between two bacteria, pili will form a tubular structure, and from one organism to the another, the fragment of the DNA will pass. Most outer layer, that is known as the glycocalyx, that is a coating of the molecules for the cell wall, that is made up with the help of the sugar, either proteins. There are two different kinds of the layer, that is known as the slime layer as well as the capsules, right? Now slime layer is loosely organized and attached to the cell wall, while the capsules are highly organized and tightly attached to the cell walls. Now consider the functions of the glycocalyx. It protects the cell from the dehydration and nutrient loss, that inhibit the killing by white blood cell in case of the phagocytosis, and contributing to the pathogenicity, and sometimes it is used for the attachment in the formation of the biofilms. See, this is the structure of the slime layer. As you know that the slime layers are not the tightly bound, so it is not firmly bound to the cell wall, and the capsules are having tightly bound to the cell wall, that cannot easily washed off by the water also. This is the formation of the biofilm. So how the glycocalyx or the capsule can be useful in the biofilm formation. So this is the hard surface, or we can say some organic surface or some coatings. First the colonies will form by the organism. This colony will produce the glycocalyx in a form of capsule. Now numbers of organisms has been produced by that colony, and that glycocalyx can be useful for the formation of the biofilm, and this glycocalyx can also use for the adherence of the other microorganism also. So here you can see that the additional microorganisms are also present in the biofilm. Archaea, that is the major domain of the prokaryotic, that is present in the extreme condition. In case of the archaea, there are major two taxa, that is Euryarchaeota and the Crenarchaeota, right? Crenarchaeota are the most thermophilic species. Example of the extremophiles are the methanogen, produce the methane. Extreme halophiles, that can grow in the extreme halophilic condition, and extreme thermophiles, that are grow in the extreme or we can say optimum temperature between 60 to 80 degrees Celsius, or the thermal vents where the temperature is more than 105 degrees Celsius. So these all are considered as extremophiles, but they belongs to the archaeal diversity. This is the hydrothermal vent where you can find the temperature 105 degrees Celsius, where you can find the extreme thermophilic organism. So this is the major comparison between bacteria, archaea and the eukarya. One more thing is there, that is the sporulation. When the bacteria are having the adverse condition, the vegetative cell converts into the spore condition, that is known as what? Endospores, right? So the composition of endospore is what? Pore, inner membrane, germ cell wall, cortex, outer membrane, inner spore coat, outer spore coat, and the exosporium. Now the main thing is what? A core that contains the DNA, RNA, proteins, right, and D-picolinic acid. Now in case of this spore, this DNA, RNA and protein can be preserved. Whenever cell will get the favorable condition, again the cell will germinate into the vegetative cell. They will produce the cell wall, cell membrane, and again these important molecules, genetic materials like DNA, RNA, protein will convert into the vegetative cell, right? So this is the sporulation mechanism. In case of that, there are a numbers of steps are there. Ultimately, vegetative cell converts into the starvation state, on-state stage, where the other energy molecules will be utilized, but the DNA, RNA and protein will be preserved in a particular format that is known as spore coat, and ultimately the spore coat will come outside, that is known as the endospore, that contains only what? DNA, RNA and protein as a important molecules. Other granules, other ribosomes are not there in a spores. So this is the particular internal structure of the prokaryotic microorganism. According to the nutritional and metabolic diversity, organism can be differentiated into the different group like phototrophs. Yes.
The difference in what conditions the cyst is formed in what conditions spores are.
When the water content is less, at that time the spore formation will be carried out. In case of the cyst, when organism wants to go in the what we can say dormant condition, at that time the cell converts into the cyst. Maybe both are the dormant structure, but generally the endospore formation will be carried out in the nutrient depletion and the water depletion. So on the basis of the nutritional metabolic diversity, photoautotrophs are there, chemoautotrophs are there. We are not going to discuss in detail this group of the organism. Third is the photoheterotrophs, and the fourth one is the chemoheterotrophic organisms are there. On the basis of energy requirement, these organisms are grouped. And the metabolic relationship of the organism, that is based on the oxygen requirement, obligate aerobes, facultative aerobes and obligate anaerobes. If we consider the ecological impact of the prokaryote, then the prokaryotics are having the different kinds of the relationship with the other organisms like a mutualism. Right, prokaryotic organisms are having the mutual relationship with the other. That the important example is symbiotic organism, symbiotic relationship with the nitrogen fixing bacteria with the plants. Right, so this is the mutualism. Then commensalism, amensalism, different kinds of the relationships are also there. So if we'll consider that there are different types of the prokaryotic organism presence on the earth. Amongst that, some of organisms are producing a valuable products, some of organisms are pathogenic, others are non-pathogenic, right? So overall scenario, we can say that the prokaryotics, without prokaryotics, ecosystem would be collapsed, because as we have studied the different cell cycles in the earlier semester, that the nitrogen cycle, sulfur cycle, phosphorus cycle, different types of the prokaryotic organisms can be involved in that cycle, right? So that geological cycles are the basic of the ecosystems, and that is run by the prokaryotic microorganism only. And ultimately, that human use the prokaryotes in the different kinds of the research and technologies, for example, in the sewage treatment, in the bioremediation treatment, in the chemical, in the medical field, and the latest research in the genetic engineering. Prokaryotic, we know that the Escherichia coli can be considered as a model organism, right? In each and every experiments, we are using what? Escherichia coli as a model organism. So prokaryotic organism can be used at any level of the research. So this is regarding the prokaryotic cell structure and the function. The major three portions are there. Cell envelope contains the cell wall and the membrane. External appendages as well as the internal appendages have their own different functions. Now there are some questions over here. Let's see, that is on the basis of the cell structure and function only. The first the question is magnetosomes in the bacteria, right? We have studied regarding the inclusion bodies or granules. Similarly, magnetosomes or mesosomes or vasosomes are there, which is the particular kind of the granule within the cytoplasm. So what is this magnetosomes, or what is the function of the magnetosomes? The options are: it helps to cell attach to the metal objects, it helps cell to magnetically attach to each other, helps a cell to float on the surface of the freshwater ponds, or it helps a cell to orient on the earth magnetic field. What will be the function of the magnetosomes out of these four? Yes? Now think over here that the magnetosomes, which bacteria contains the magnetosomes, or from where we can found the bacteria which contains the magnetosomes? ऐसे कौन से ऑर्गेनिजम्स है, ऐसे कौन से बैक्टीरिया है जिसमें मैग्नेटोसोम्स हो सकते हैं? It is not B that is magnetically attached to each other. It is what? It is D, that is what? It helps cell to orient in the earth's magnetic field, because those organisms are having the magnetosomes. They are widely isolated from the deep sea, deep of the oceans, right? So because they are having the magnetosomes, they are having the higher attraction to the earth gravity. So the major function of the magnetosome is what? Helps cells to orient the earth magnetic field. The second one is the 70S prokaryotic ribosome consists of what? 70S consists of what? B, that is 50 and the 30, right? So what about the eukaryotic? 80S, 60 and 40, right? Now another question is, a gram-positive cell have a second outer membrane that helps retain the crystal violet stain, have a multiple layer of the peptidoglycan that help retain the crystal violet stain, have a thick capsule that traps the crystal violet stain, or have a periplasmic space that trap the crystal violet. That is what? Yes? B. That is a B. That is gram-positive cell have a multiple layer of the peptidoglycan that helps to retain a CVI complex. The next question is, the presence of D amino acid in the cross link of the peptidoglycan layer is most likely because most peptidase can only cleave L amino acid. D amino acid fit the structural constraint of the cell wall better than the L amino acid. Most L amino acids have already been used for the protein synthesis. Or D amino acids are easier to cross link in the absence of the ribosomes. Yes? The presence of D amino acid in the cross linking of the peptidoglycan, N-acetyl muramic acid and N-acetyl glucosamine monomer, when form the polymer for the peptidoglycan layer, at that time, which will be the option? This is what? A. This is what? Most peptidase can only cleave the L amino acid. The next question is, the third amino acid in the peptidoglycan cross-linking chain is either diaminopimelic acid or lysine, because this amino acid must be positively charged for a salt bridge to form, be hydrophilic, have a free amino group for a peptide bond formation, or have a large R side chain for fill a space in a cell wall. We have discussed the 4 amino acid in the monomer of the peptidoglycan, no? Amongst that, the third amino acid was what? Diaminopimelic acid, right? It is C, that is have a free amino group for the peptide bond formation. The next question is, the gram-positive cell wall have a thick homogeneous cell wall, have a large amount of the teichoic acid, do not have an outer membrane, or all of the above. It is what? All of the above. The outer membrane of the gram-negative cell is more permeable than the plasma membrane. Why? Because lipopolysaccharide is larger than the most membrane phospholipids. Lipoprotein stretch the outer membrane. Porine protein establish the hole in the outer membrane, or the core polysaccharide spans the lipid bilayer. Which condition is responsible for the permeability of the plasma membrane, outer plasma membrane of the gram-negative cell wall? That is what? C. Because the outer, as we have discussed, when the gram-negative membrane contains the outer and inner. In case of the outer, there are a particular type of the protein that was known as porine protein that can allow to enter or exit a particular kind of the molecule, that is porine protein, that establishes the holes in the outer membrane. The most important role of the prokaryotic cell wall is to maintain the shape of the cell from the osmotic pressure, prevent the ion from the diffusing away from the cell, or block the effect of antibiotic like penicillin. The most important role of the prokaryotic cell wall: protect the cell from the osmotic pressure, or the maintain the shape of the cell, or the prevent the ion from diffusing, or the block effect on an antibiotic. It is B. That is what? Protect the cell from the osmotic pressure. That's why the gram-positive organism can tolerate the higher osmotic pressure than the gram-negative organism, and that's why they can survive even in the depletion of the water instead of the gram-negative microorganisms, because of the thick layer of the peptidoglycan cell wall. Which of the following is not true about the capsule and slime layers? Not true. They consist of a secreted material lying outside of the bacterial cell wall. They can prevent desiccation of the bacterial cell. They are required for the bacteria to grow normally in the culture, or they help bacteria resist phagocytosis by the macrophages. Not true about the capsule and slime layer. That is what? That is not required features for the growth of microorganism. That is independent characteristics of the microorganism. The fimbriae attach bacteria to various surface, cause the bacteria move through the fluids, sense changes into the nutrient concentration, or fimbriae are a pathway for the secretion of the exoenzymes. Which is true for the fimbriae? Attach bacteria to the various surface, cause a bacteria move through the fluid, sense changes into the nutrient concentration. Yes, it is A. That is used for the adherence of the bacteria into the any solid surface. So this kinds of the question can be asked in the examination as a multiple choice. But when we are discussing regarding the cell structure and the function, the biochemistry is the most important portion, because the major question has been covered from the biochemistry instead of the functions, right? So you have to give the more weightage for the portion particularly of the biochemistry. Thank you very much all for listening me patiently, and this material has been prepared from the internet, various sources of the internet. So I like to thank all the person who may ever concerned with this material. Thank you very much.